Hello, colleagues. Those who are in Zoom, hope you can hear me. So let's start today's topic. We will need to talk about three very large slides: eye development, then dog's head slide, and rat embryo slide. So these are quite big ones. So I will quickly turn on everything here, and then we will start checking different slide variations. We will first start with eye development. So first slide will be eye development. So just wait a moment. Those who are in here, again, I will pass around the paper so you can write down your names. So today is 8th of May. And 1, 2, 3, 4, 5. So write down your names again on the paper. Yeah, not just to check the outside attendance. I hope you have a pen. Maybe this will work. That's right. Okay, so let me turn on the camera also. So, and then we will start checking different slides. So I will share the Zoom so everything is visible to you. So first, eye development. There's different slide variations. In this case, you don't need to remember the weeks because different types of eye slides. Some slides are from humans, approximately week 11, 12. Some slides are from animals. So in this case, the week numbers are not that important, but there's a lot of structures to check. So we will go through all of them so everything is clear. Again, I will put side by side images. So I will have the picture of the slide on one side from these to these. And on the other side, we will have the eye. I will hide this and we will check the slide in detail. So, first of all, what we need to check, I will move the camera so it's a bit bigger, and the picture. First things first, you see the developing palpebra. So you have the eyelid or the palpebra here. So developing eyelid, that's number one. So developing eyelid. So you see the eyelid that's growing, developing here. So developing eyelid. So number one, developing eyelid. And because it's the eyelid, what you should notice here. So first of all, you have two surfaces, like in histology. One surface shows the developing epidermis. That would be number two. So developing epidermis. So developing epidermis. That's number two. And on the opposite side, so epidermis is 2, on the opposite side, you have the developing conjunctiva. So this conjunctival surface or developing conjunctiva, it develops on the other side. So number 3, developing conjunctiva. What else is here? So if we have epidermis, below the epidermis, you have dermis. So number 4, that would be developing dermis. So number 4, developing dermis. So developing dermis is also present. So developing dermis. Then in the middle of the eyelid, you can see skeletal muscle tissue, both in the picture as well as in the slide. You have developing skeletal muscle tissue. So this is developing skeletal muscle tissue, but you also can use another name, musculus orbicularis oris. So developing musculus orbicularis oris. So for these slides, that's also quite specific detail. So inside, in the middle of the eyelid, muscle tissue is developing. You can use the terms from anatomy and histology, like developing musculus orbicularis. Oculi. I will delete this. I will rewrite everything. So number 5, developing skeletal muscle tissue, and muscle is musculus orbicularis oculi. Sorry, automatically remembered another name. Oculi, the eye is oculus, that's why. So musculus orbicularis oculi. What else here? So there's a specific area. Here, number 6, that's an embryological structure. You can see that both eyelids are closed shut, so there's epithelium, which is proliferated in between. So number 6, proliferated epithelium. Proliferated epithelium, that's number 6. So the epithelium grows in between the eyelids and connects them together. And later it degenerates. Later during second trimester, so this epithelium degenerates and eyelids can open up. So proliferated epithelium. There's some other names in some textbooks. You might see eyelid epithelial seam. That's also a name that's used, but proliferated epithelium is more than enough to remember. Next question to you. You can spot hair follicles here at the tip of the eyelid. What do you think? What are these hair follicles? Eyelashes. Eyebrows are above the eyes, so those are eyelashes. So 7 eyelashes. Yes, eyelashes, developing eyelashes. Eyelashes. I will delete. Right, correctly. Developing eyelashes. So eyelashes are here. If you see hair follicles at the tip of the eyelid, eyelashes. What else? Then here you see a cavity. There's a cavity behind the eyelids. And this cavity, we call it the conjunctiva space. Conjunctiva space. Number 8, conjunctiva space. Conjunctiva space. Or conjunctiva sac. Also, you can use conjunctiva sac. Conjunctiva sac. It means the same thing. It's a cavity behind the eyelids. In your e-studies written as eyelid space also, technically it's a synonym, but better use conjunctiva sac. So in your e-studies, some descriptions, there's another name used, but conjunctiva sac is the most correct name here. Then, next, if you know eye anatomy, well, this slide won't cause you big problems. First, let's check here. When we start to check the eye, we see there's a plate of tissue here. With number 9, we see developing cornea. So developing cornea. So developing cornea, the anterior portion of the eye. Layers of the cornea won't be asked in the colloquium. So layers, like in histology, are not that well developed. Some are developed, of course, but these layers won't be asked in colloquium. Just remember developing cornea. Bless you. Then, behind the cornea, you see a space. So there's an opening here. And this space, we call it the anterior eye chamber. So anterior eye chamber. It's a space located behind the cornea. It's filled with a fluid, aqueous humor. So this anterior eye chamber just behind the cornea. Anterior eye chamber, that's number 10. Then you can spot here there's a very thin membrane, number 11. It is an embryological structure. This is the generated membrana pupillaris or pupillary membrane. So Number 11, degenerated membrana pupillaris. In your studies, partly degenerative membrana pupillaris, if you remember degenerated membrana pupillaris, it will be okay. So it's a very thin membrane that quickly disappears. So when it dissolves, the pupil is opened and it allows light to move deeper into the other eye structures. But in some people, this membrane can persist and it can cause visual disturbances. If there's additional membrane, so it's harder for light to pass deeper into the eye to reach the retina. So persistent membrane pupillaris is an interesting pathology. You can Google it and you can see how the eye actually looks like if it has this pathology. So it's associated with the persistence of this membrane. Okay, next, a bit of a trickier thing. So here I will enhance this area. So with number 12, and in slides that would be here in the pictures, this is developing iris. So number 12, developing iris. That's the colorful part of the eye, iris. You can notice that at the back of the iris, there's pigmented epithelium, while in the front, there's no epithelium at all. So iris has this duality in its look. So you have on one side epithelium, but in the front end, there's no epithelium at all. So iris, developing iris. And then a very tricky structure. Behind the iris and behind the membrana pupillaris, there's a very small cavity just behind the iris. I will use the opposite iris here in the picture. And here I will also label below here. So number 13, this is posterior eye chamber. Posterior eye chamber. Posterior eye chamber. So it's a very small cavity just behind the iris and it's in front of the lens. So it's a very small cavity. It's not visible on every slide. In this slide, we can see it. In most slides, it won't be visible that well. Then what else? Here in the slide, I will, sorry, that's number 13. Yeah, 13, 14. You see there's this corner here. So this corner area, this whole area here in the corner. So in this corner, you have corpus ciliare. So developing corpus ciliare or ciliary body. So developing ciliary body or corpus ciliare. In your studies, incompletely differentiated corpus ciliare, but it's a longer name. If you just remember developing corpus ciliare, ciliary body, that's okay. So it's an area that has a lot of smooth muscle tissue. And another detail, you can spot there's some processes coming from the ciliary body, some thin, small processes. With number 15, we call these ciliary processes. So developing ciliary processes. So developing ciliary processes. So these processes produce the eye fluid. Eye humor or aqueous humor is produced by ciliary processes. So when they are positioned towards the posterior eye chamber. So ciliary processes. Okay, so far so good. What else is here? Here in the slide, you can spot this whole structure. This is the lens. So lens, developing lens. So lens. So developing lens. So that's this whole structure. So developing lens is all of this. So that's the lens. So developing lens. Maybe I will do it a bit differently. So the lens is all of this. But inside the lens, there's some additional details, some additional structures that are visible in high magnification. These following structures typically are not always visible, but I can label here so that everything is clear. First of all, you can see that in front of the lens, there's an epithelium. So this is anterior lens epithelium or anterior cuboidal lens epithelium. So 17, anterior cuboidal lens epithelium. So this epithelium is in front of the lens. In the big picture, it's very hard to show it. It's just at the very surface of the lens. So the anterior surface of the lens has this simple cuboidal epithelium. So anterior lens epithelium. From this epithelium, very long cells in the lens will grow. And in the microscope, if you zoom in, you will actually see very elongated cells inside the lens. These long cells with number 18, these are called lens fibers. So 18, lens fibers. Lens fibers. So these are very long cells that fill the lens in. So lens fibers, so very, very long fibers, very long cells. In the microscope, in this picture, it's quite hard to show these lens fibers, but no, technically they're also here. So lens fibers, so very long fibers, very long cells inside the lens. As we get older, the amount of lens fibers increases and the lens becomes more dense. It's harder for it to change the shape, and because of this, so there can be some problems with accommodation. So as we get older, the lens is less elastic, so it's harder for it to round up, and it's more difficult to focus on objects that are close to our eyes. So that's why glasses are needed typically as we get older. 40s, 50s, 60s, glasses for reading are typically needed because of this. What else? You can see that behind the lens, you have a gel-like substance. Now I will point here, this whole area with number 19, this is corpus vitreum. So 19, corpus vitreum. Corpus vitreum. So corpus vitreum is this gel-like substance that fills basically the whole eyeball. And in the corpus vitreum, normally in adults, there's no blood vessels. But during embryonic development, you will see small blood vessel fragments, typically close to lens. So these blood vessels are remnants of the arteria hyaloidea, remnants of hyaloid artery. So remnants of arteria hyaloidea. This is a structure. It's not visible in all slides, and it's additional trickier structure that's sometimes asked. So this is an artery that provides blood supply to the lens, but only during embryonic development. After the lens has developed, these blood vessels disappear. So they completely degenerate. And here, so these remnants of blood vessels can still appear during embryonic development. If they persist for longer, they again can cause visual disturbances. So normally there shouldn't be any blood vessels in the corpus vitreum. Okay, is this part of the slide understandable? That's the front part of the eye. Now we will move to the back part of the eye. So I will clear the drawings. Here, if you want to take a screenshot, take a screenshot. I will move to a different location. If you don't want to take a picture here, I will move on. So I'll clear all of these off. I will move to the back part of the eye, and we need to talk about some other layers and structures. Now I will clear all of this, quite a lot of drawings here. Okay, so in the microscope, again, I will move to the back part. In this region, approximately. Or we can also do it here, doesn't change a thing. So, if we move here at the back of the eye, what you should notice here. So you might see some elements. For example, you see there's a layer of connective tissue that surrounds the eye. That's number 21, developing sclera. So developing sclera. That's the connective tissue that surrounds the eye from the outside. So developing sclera. So 21, developing sclera. Developing sclera. I will move this text box here. So developing sclera. And below the sclera, you again see a layer of connective tissue, which is similarly staining to sclera. This thin layer of tissue, which has quite a lot of small blood vessels, developing choroid or developing choroidea. So 22, developing choroidea or choroid. So sclera is on the outside. The next layer, the choroid, is already developing. It's not yet pigmented, so blood vessels start to appear here, but it's not well developed yet. And then the tricky bit. On the surface of the choroid, you see 23 pigment retina. So pigmented epithelium, you can only see it clearly in high magnification, but 23, this is pigment retina. So this pigment epithelium of the retina sits here, and here, 24, this is neural retina. So you have pigment retina on one side and neural retina on the other. So what's interesting here? So pigment retina is just this pigment epithelium. It's not connected to the other layers of retina. You do not need to distinguish the different sublayers of the retina like in histology. You just need to remember pigment retina, neural retina during embryonic development. Here you see there's a big gap in between. This opening, or it's basically an artifact which we call the intraretinal gap. So 25, intraretinal gap. Intraretinal gap. Again, I'll rewrite this now. Intraretinal gap is an artifact that appears in slides. Intraretinal gap. Yeah, no, it's correct. Intraretinal gap. The thing is, pigment retina, it's not tightly connected to the neural retina. There's no cell junctions at all. And for example, if this intraocular pressure is decreased, or the eye experiences trauma, neural retina can easily detach itself from the pigment retina. This is called retinal detachment, and it's quite bad, as you understand, because if it's prolonged, it of course can cause blindness, because neural retina can eventually die if it's separated too long from the other layers of the eye. So this is a typical problem in typically eye trauma, retinal detachment. During embryonic development, we see during slide preparation, as the tissue shrinks, the neural retina typically detaches itself from the pigment retina. This is one of the rare cases where we ask an artifact in slides. Intraretinal gap, remember this. There's some clinical significance to it also. What else is here? In this slide, in this corner with 26, we can spot here skeletal striated muscle tissue. Again, but these are also called extraocular muscles. These are the muscles that help to move the eyeball around. So 26, developing skeletal striated muscle tissue, and you can call them extraocular muscles. In the slide, so it's eyeball muscle differentiation, but you can use also this term. So developing skeletal striated muscle tissue or extraocular muscle development. So if you see muscle tissue outside the eye, most likely these are extraocular muscles, if they're connected to the eye, of course. Then let's move to some special areas in the slide. I will clear these unnecessary areas. I will move to some other areas. So if we move to a different location here, so just above the eye, you might spot some other tissue, like here. You can spot bone tissue. So this is orbit bone development. Here, 27. So developing orbit bone or intramembranous ossification even, so or bone development, it will be okay. So bone development can appear in slides. So 27, bone development. And in the eye region, it's interesting. You can have both intramembranous ossification and endochondral ossification. It depends on the slide and the location. You might even see cartilage. So in this slide, you see bone tissue, but we will check some other slide pictures. You might see cartilage even there. So, and let's move to this bottom part of the eye, 28. So number 28, here, this bottom region, I will move also in the slide here. I will clear unnecessary drawings here. Here you see some fragments of glandular tissue. This is a developing lacrimal gland. So developing lacrimal gland. So it's special for this slide that you might even see lacrimal gland development. So developing lacrimal gland, it appears in some slides. So developing lacrimal gland. About some other variations, we will go through the pictures because not all of them are on the slide plates, but you can check them on your own. So we will go through some of the slide variations now. So if this slide is clear, you can take a screenshot. If not, you also can quickly check it and write it your notes, but that's the idea about this slide variation. Okay, if this is clear, let's check other eye slides. So I will clear here. Let's check some other slide variations now. I will enhance the size of this slide. Okay, let's check some other slides. This is the same slide as the first one, only it's turned to the side. So it's basically the same slide. I won't repeat the structures. Then there's this slide. So let's go through this one. I will ask questions to you also, those who are in the chat and those who are on site. I will ask questions also. Let's choose a color that stands out. So first of all, what is this structure, number one? Lens. So developing lens. So eye development, one, that's lens. So developing lens, good. That's number one. What is behind the lens? Corpus vitreum, good. Corpus vitreum development. Corpus vitreum, good. What's further here, number three? Neural retina, good. And here on the surface, yes, on the surface, if only the surface is as pigment retina, only the surface, that's why I will put an arrow tip here, only in the surface pigment retina. Okay, what do you think, what's here in the front, number 5? Yes, developing iris. So developing iris is here. And what comes here? Number 6, you see small fragments of tissue going out. What are these? Yes, developing ciliary processes. Good. What is here? Number 7. It's this plate here. What is this? Not sclera. What is in front of the eye? Cornea. Yes, developing cornea. Yeah, thank you. Developing cornea. Good, thank you. Then, what is this gap here? Number 8, in front of the cornea. No, in front of the cornea. Behind you have the anterior eye chamber, but in front of the cornea you have conjunctival sac. That's the tricky bit here, conjunctival sac. And now the problem. The problem is all of this here is the eyelid. So number 9, all of this is the eyelid. So eyelid, developing eyelid. And you see there's a big artifact. The epithelium, this conjunctiva, has fractured from the surface of the eyelid. That's why you see another big opening. It's an artifact. And it can cause some problems. It's hard to understand where the cornea is. That's why be careful. This slide can also appear in the colloquium. So be careful. Typically, I'm not that able to ask in this location, but sometimes it can happen. So be careful with this, with these slides. So where these crosses, that's a big artifact, and the eyelid is there. You can even spot here with number 10 this proliferated epithelium. So of the eyelid, proliferated epithelium. So proliferated epithelium. So proliferated epithelium. Technically, some other structures can be found here, like the eyelashes technically can be found as small fragments, muscle tissue. One thing that I will emphasize here with 11, in all of these locations where I put number 11, there's bone tissue, so developing bone tissue. Developing bone tissue. And interestingly, it's only in this slide, this small fragment of tissue, I will put an asterisk, 12 with an asterisk, hyaline cartilage. This is one of the slides where there's hyaline cartilage fragments. Small hyaline cartilage fragments can appear. In this slide, due to the magnification and staining, it doesn't look that great. It's very blurry, but if you would check this with your own eyes at the microscope, you would see hyaline cartilage. This slide, unfortunately, is not on the slide plate here, or we'll need to recheck at the end of the class, but this is a small hyaline cartilage fragment. It can appear in some slides. Okay, this is just a variation. Let's check some other slides. I will clear these drawings, some other pictures, like these ones. So there's some structures that appear only in a slide like this, like here, developing optic nerve. So here, developing optic nerve. So it can appear in some slides. It's not in all slide variations, so it's quite rare. And again, all the structures can be logically determined. You have the lens here, so lens is visible nicely, cornea. Neural retina, we'll label as NR, neural retina, pigment retina, corpus vitreum CV, so corpus vitreum. So all these structures repeat again and again. You can even see small eyelid fragments, but what stands out here with the green color, you see fragments here. Now I'll put number 2. All of these are hyaline cartilage. Hyaline cartilage. There's small cartilage fragments. This is one of the slides where there's no bone tissue, but you see cartilage next to the eye. It can appear in some cases because chondrocranium is also quite close, and some orbit bones also develop from cartilage directly. But also desmocranium can be close, so it's a unique location. Yeah, but if you see hyaline cartilage, this is how it typically looks like. That's the general idea here. Then there's some other variations. Now this one is a tricky one. It's very similar to the previous one. So it's from the same type of slide, but I will emphasize the tricky part. I think the lens and the cornea is clear. Neural retina, pigment retina, and all the other layers should be understandable. But the tricky part is here. What is happening here in this area? So this is a place where part of the eyeball wall has been cut tangentially. What it means? So here you see pigment retina, and there's pigment retina also here. So pigment retina. You see both in both areas fragments of the pigment retina because part of the eyeball's wall has also been cut. And the tissue inside here, this is the choroid. Now I will put CH, choroid, choroidea. So I will write down, so PR means pigment retina, and CH means choroid. So here, this is a tricky section because you see also a fragment of the eye wall, and also neural retina here can be fragmented. These are fragments of the neural retina. So all of this is neural retina. Here, all of these neural retina fragments. Neural retina. Neural retina can sometimes mimic the lens. What I mean by this, I will open the pictures from the last week a little bit also. Last week we had, that was 5th class, here. Last week, remember not last week, 2 weeks before that, brain development, this slide, for example. We had this slide here. You also see eye development, but you might see a fragment that looks like this. That is not the lens, that's neural retina. Neural retina can look like a round fragment inside the eye. And position is important. If you see something round at the back of the eye, it's definitely not the lens, it's most likely neural retina. So from the last week's slide, we can also see it partially. That's why I'm showing this. So in eye development, sometimes the neural retina can mimic the lens, but it's not the lens. It's made from tiny dark basophilic cells. So that's the tricky bit in this slide. Okay, about the smaller structures, I won't repeat them because we have 2 more large slides. Any questions? Yes. Eye development. So slide name here for all of these eye development. Eye development, it's completely fine for this slide. Then another just for fun. This is homework for you. Basically, you repeat eye anatomy. It's almost a completely developed eye, and you can check all the layers. Here, even the retina is fully developed. And here, this slide is also used in histology. So this is more of a histology slide. Most slides in our slide plates are not like this. They are typically during embryonic development. Then, that's the ear. That's another story. This one. So if you cut the eye at the back side, so you don't see the lens at all, but you cut the eye approximately here in this region. If you cut it through here, then you will see something like this. So basically, the slide shows the following. You have the optic nerve. So optic nerve is in the middle. Neural retina, intraretinal gap, and pigment retina. So here you see the back side of the eye. There's no eyelids, no lens, nothing like that. You only see the back portion of the eye. So this is a very tricky slide to check because you don't see other eye structures. But if you think logically, it's not that bad. Of course, technically, we still have the choroid and the sclera, the connective tissue outside. Sclera, choroid and sclera, but it's not always visible that well. CH, that's choroid and sclera. But that's the general idea here. Okay, that's everything about eye development. So we have talked quite a lot. Next, let's check some special cases. So this slide is a very interesting one. So This slide, technically it's eye development, but there's more details to check here. So here, as you understand, that's the eye development. So eye development is here. I will just write eye development. So eye development. Here you can see the lens very well. Neural retina stands out very good. You even see the cornea here in the front. And some other structures, but that's the general idea. So you see the eye. Then there's some extra structures. Here, this shows developing nasal cavity. So developing nasal cavity. And there's a couple of structures to label here. I will write numbers here and we'll use different color. Red stands out good here. So you see here, that's number 1 here. That's developing nasal mucosa. Developing nasal mucosa. These slides are taken from animals, from rats mainly. There's also dog slides, but we will check them separately. So this slide is a special case where next to the eye, we might see some additional structures. Then here you see hyaline cartilage. And this hyaline cartilage, it's called nasal cartilaginous capsule. Sorry, yeah, so the question from Natalie, is it the dark or light area included? So in the nasal mucosa, both the dark and the light area together. That's nasal mucosa here. And the cartilage, so these cartilage plates and cartilage fragments, they form nasal cartilaginous capsule. If you remember developing nasal cavity, that's also okay, but it's the hyaline cartilage that will form parts of the nasal cavity wall. So nasal cartilaginous capsule. Then, what else? I will encircle this whole area, and here you can write ear development. So ear development, so the ear develops here. And in case of the ear development, some structures repeat from the previous weeks. Like here, if you see this hyaline cartilage, that's ear cartilaginous capsule. So ear cartilaginous capsule. Ear cartilaginous capsule. So, and inside you have the developing inner ear. So, developing inner ear, or you can write cochlea. So developing cochlea, developing inner ear. If you see membranes arranged in groups like this, so the inner ear is developing inside. That's the general idea for this slide. And next, we can move on to the dog's head slide. So dog embryo's head sagittal section. That will be the next one. I will check what is in the slide plates because there's some limitations. We will check these slides. I will check the slide plates. Most likely I have rat's head here, but rat's head slides are quite limiting. Here we got lucky. We have a dog's head. Good. Okay, again, I will use the pictures to go through different areas here. The tongue is here. Okay. So, first of all, so this slide that I'm showing here, so that's dog's head sagittal section, or dog embryo's head sagittal section. Or you can also describe this as a development of semicircular canals. That's another name that's also in the slides. Development of semicircular canals, dog embryo's head sagittal section. So, yes? Yeah, that also works. Head development, dog embryo also works. Yeah, that also is okay. So, I will use, again, camera on one side, slide on the other side. So this is quite a big slide, so we will carefully go through different areas. So, first of all, I will start with this region here. Now I will use green color. Let's start here. Eye development. So eye development, that would be this whole region. So eye development, that will be number one, eye development. I will move this also on the camera. So eye development, approximately in this region. In this dog's head, actually, the eye is not present at all. There's only a small fragment from the optic nerve. So, in different dogs' head slides, there can be variations. Here, the eye development is super small. You only see the optic nerve, but on the slide picture in your e-studies, you see something bigger. Interestingly, here with number 2 in the picture, only in the picture, but not in the slide, neural retina. So number 2, neural retina. So neural retina is visible. Neural retina. There's no lens here, only neural retina. That's special here for this slide. Then let's move to this area. I will encircle this whole area with number 3, developing nasal cavity. So 3, developing nasal cavity. Developing nasal cavity. In the slide, that would be here. In this upper region, you see some hyaline cartilage fragments. This slide is not exactly the same as in the picture, but it has some similarities. And here in this developing nasal cavity, if you see hyaline cartilage, like here in the slide, again, you can use nasal cartilaginous capsule. Nasal cartilaginous capsule, this is the name that's used sometimes. Or if you just write nasal cavity development, it will also work. But sometimes if for this cartilage, you use the specific term. Then, what else? In front of the dog's head, with number 5, you see hair follicle development. So developing hair follicles. So developing hair follicles. Here, if you move to the front of the dog's head, you will also might see some fragments of hair follicles here on the top region. Here in the slide, we just a little bit can see some fragments of hair follicles. So in different locations, you might have hair follicle fragments. So developing hair follicles. Then here, number 6, tongue development. So 6, tongue development. Sorry. So tongue development. In the microscope, that would be here. You see, there's a bigger fragment of the tongue in the microscope. In your e-studies, you only see a small fragment of the tongue. It can have variations depending on the slide. And then what's interesting here, on this upper jaw region with number 7, late tooth development. Late tooth development. Late tooth development. And late tooth development can also appear in the lower jaw. Check carefully where I'm encircling this. Number 7 is both in the upper jaw and the lower jaw as well. Late tooth development. if you check the slide, you might see this late tooth development. So the developing tooth here. Typically, late tooth development structures won't be asked that much, but what can help you sometimes? You see this light blue structure. That is dentin. So dentin is light blue. In these slides, this is Mason's trichrome. Dentin stains blue. And this cherry red or purple structure, that's enamel. So you can spot dentin and enamel, but only if it's zoomed in. So dentin and enamel can be seen here. Dentin is blue, enamel is cherry red. So dentin and enamel is visible in the slide. So dentin and enamel. Yes? This is the smallest magnification. The slide, yeah, this is the smallest zoom possible. So these are tricky slides because of their size. These are one of the biggest slides we have. Yeah. Okay. What else? That's one detail. I will clear this a little bit. Then what else? I will move to the lower jaw a little bit. Sorry. In the lower jaw region, also sometimes in the upper jaw, you might see a lot of glandular tissue. So we will move to these lower parts. That's basically this region here. We will zoom in here. And what we will talk about. So we will need to talk about glands. So there's multiple glands here. Here, I will encircle this one with number 8. That's developing sublingual gland. Then below with number 9, developing submandibular gland. And number 10, brown adipose tissue. I'm telling you the terms, and then we will zoom in and check how they look like actually. So sublingual gland, submandibular gland, that's number 9, and brown adipose tissue. So this neck region is very tricky because all of these tissues at low magnification look very similar. So here in the microscope, I will zoom in a bit closer so you can see the differences. So if you zoom in higher magnification and you check these glands here, so there's quite a lot of them. They look quite similar to each other, but some glands look a bit lighter. There's even some mucus tubules visible, so light staining cells. That is how the sublingual gland looks like. If you see some mucus cells, so light staining cells, this is how the sublingual gland looks like under high magnification. If you want to take a screenshot, please take this because it will be very helpful for you in the colloquium. So you need to see some mucus tubules, then that's the developing submandibular gland. Those other structures will be asked inside, just need to remember what is this gland here. Sublingual. It has mucus tubules. Sublingual, glandulus sublingualis. Sublingual gland. So high magnification of sublingual gland, you should see mucus tubules. So mucus tubules are visible. That stands out here. Sublingual gland. That was number 8, if you zoom in. If you would zoom in into the submandibular gland, it has no mucus tubules yet. It's very dark staining. Submandibular gland is very basophilic. So it's very basophilic, no mucus tubules at all in the slide. Submandibular gland looks like this. And in many slides, it's the only gland. In rat slides, it's the only gland. If you have the rat's head, it's the only type of gland that you will see. But submandibular gland is very basophilic, no mucous tubules. And finally, in high magnification, brown adipose tissue. So when you check brown adipose tissue, you will see lipid inclusions. They can be bigger, they can be smaller, but small lipid inclusions. So this is how the brown adipose tissue looks under high magnification. Brown adipose tissue, you see lipid inclusions. So it has this granular appearance. So it's really grainy granular because there's a lot of small lipid inclusions. Yes? Larger spots could be white adipose tissue also. So white adipose tissue can be mixed with brown adipose tissue. That's why you see big white spots. Those most likely white adipose tissue cells. White adipocytes, yeah. But brown adipose tissue looks like this. So again, I will repeat. So brown adipose tissue, this is how it looks in high magnification. Submandibular gland. And further out with mucous tubules, sublingual gland. The good thing is about the glands. So if you accidentally miss the sublingual gland with the submandibular gland, it won't be a big mistake. You can get a full point because it's very difficult. But please don't mix the glands with brown adipose tissue. So don't mix that. And another thing, number 11 here, skeletal muscle tissue. So if you move through the neck region of this dog's head, you will see skeletal muscle tissue. In high magnification, you will see fibers. But in some slides where you have cross sections, you can easily mix it up with brown adipose tissue or the glands. I will check here. For example, here, if you have a cross section, you can easily misinterpret this with glandular tissue. Be careful. Skeletal muscle tissue also can look very similar. So skeletal created muscle tissue also appears. So number 11, you see skeletal muscle tissue in high magnification. Okay, then what else is left? We need to check the developing ear region. So that would be here, this area. So what is interesting here? This slide has a very well-developed cochlea. So approximately in this area, I will put number 12. So this region, it has developing cochlea. So number 12, developing cochlea. We can also see this in the slide. So developing cochlea or developing internal ear. Here you can even see organocorti, basically the same structures that were in histology can appear here a little bit. But what's special here? If you move a bit further, here we'll put number 13. We have the ear cartilaginous capsule. So ear cartilaginous capsule. So basically, if I move to these parts further away and if I see hyaline cartilage, that's ear cartilaginous capsule. So hyaline cartilage. In the slides, a bit different. So this light blue structure, yes, this light blue structure, that's ear cartilaginous capsule. Ear cartilaginous capsule, this whole hyaline cartilage, that's light staining in the slide. So ear cartilaginous capsules, light blue in the stain typically. There can be some bone tissue already. In this slide, ossification is already happening. Bone tissue also appears. But what is interesting, I will use a bit different color, only in the slide picture. In the microscope slide, we don't see it that well. You see these rounded openings here, here, also partially here. These are developing semicircular canals. Number 14, developing semicircular canals. These openings, round openings inside cartilage, developing semicircular canals. So you can see these developing semicircular canals. There's a lot of structures in the slide descriptions that are also mentioned, but in the slides we cannot really see that well. For example, organ of Corti is not in the slide picture that well, it's not that well visible. We also don't see ganglion spirale. We don't see the temporomandibular joint also. So some of these structures not really visible. Technically, they could be found as small fragments, but these are not the structures that would be asked in the colloquium. What else is here? Again, I will use the green color. You see there's a plate of hyaline cartilage, number 15 here. Chondrocranium. Chondrocranium. And in case of the microscope, chondrocranium would be here. You see another plate of hyaline cartilage that's a bit further out. Chondrocranium. So that's hyaline cartilage here. Chondrocranium. And if we have chondrocranium, we also have desmocranium. 16, desmocranium. I wrote that in the slide picture. But we will see desmocranium here at the very top of the head. So desmocranium, this blue plate of bone tissue, desmocranium. Desmocranium. So desmocranium is visible here. So this is also a specific slide variation here, desmocranium. This blue staining part, that's, yeah, desmocranium is this light part here. This is desmocranium 16. Yes, above skin development, so developing skin. There's epidermis development, hair follicles, dermis development, like in the 4th class when we talked about these topics. Skin develops on top. Hair follicles, the epidermis, dermis development. Okay, some other elements, some structures that are not visible in all slide pictures. Sorry, let's return this back. What else is here? Only in the slide picture, you might see brain development. So there's small brain fragments. Which parts of the brain is hard to say, but sometimes you might see fragments of the brain. Brain development. Brain development.
Professor?
Yes?
Yeah, okay. You just wrote it down. I thought number 17.
Yeah, 17.
Yeah.
There's a question from Karina. Hair follicles are these small structures here. These are hair follicles also. And also at the tip here. So at the front part of the dog embryo also see hair follicles. But hair follicles are in different locations all around the embryo. Hair follicles. Okay. We have one more slide to check. If we have time, we will check some other slide variations, but we need to move on to the rat embryo sagittal section. Before we check the rat, very briefly about some of these slide variations. So for dog's head, there's some variations that look like this. Not this, sorry, oops. So there's a variation that looks like this, very similar to the first one. Only thing is, you don't see the oral cavity. Tongue is very small. You have a very small tongue fragment. And technically, upper jaw moves to this side and lower jaw is towards that side. So the tongue is very small. You don't really have a nice big oral cavity. But again, you have the eye development. You see the neck region with all the glands. Here, there's a small sublingual gland. Sublingual gland is here, so it's very small. Small fragments of submandibular gland. and brown adipose tissue. Brown adipose tissue in large quantities. Inner ear, same idea. If you see round openings inside hyaline cartilage, developing semicircular canals. So developing semicircular canals are all of these small openings. Semicircular canals, SC, semicircular canals. I don't have the slide on the slide plate. Most likely it's on the top floor or some other plate, but this is also a variation of the dog's head. Okay, we need to move on. We need to talk about rat embryo total sagittal section. So, about rats. So some rat slides are given just like a small fragment of the head, but we will check these ones, these very big slides, and go through different parts of the rat's embryo. So I again will put the microscope on, and we will go through the rat's embryo slide. So let's take this one. So let's check here. So these slides are very big. This is the lowest magnification. And typically when we talk about the slides, we go through different zones. So first, we are checking zone that's located in a very specific place here, where the oral cavity starts. So no, I don't need a text box to delete. Okay, I will use this. Here, we will start here. So this is the lowest magnification. So you can notice the slide is gigantic. So this is one of the biggest slides that we have. But we will start first. Number one, tongue development. So tongue development. So slide name, rat embryo sagittal section. And one, tongue development. When we talk about the tongue is quite large. It also has developing tongue papilla, muscle tissue like the tongues we saw in the 4th class. So I won't repeat all the structures inside, but that's the tongue development. Then, as you understand, you have the upper lip and the lower lip. In this slide, that would be like this. So two upper lip, developing upper lip and lower lip. So two upper lip development through 3 lower lip development. So you see both lips, upper and the lower lip. We will move into the upper lip. So we'll check the upper lip region. Upper lip. So when we move into the upper lip, you might see late tooth development. So here with number 4, this is late tooth development. And here in the slide, you can see it here, all of this late tooth development. Late tooth development. Sometimes there's no teeth at all. There are slides where you don't see any teeth at all, only in the lower jaw or none of the jaws. But in some slides, you might see teeth, late tooth development. Then, what else? Let's move a bit further. If we move in the upper jaw, you will see developing nasal cavity. So nasal cavity development. And again, where we see the cartilage with number 5, that's ear cartilaginous capsule or nasal cavity development. So where there's hyaline cartilage, that's number 5. That's nasal cartilaginous capsule. Oh, sorry. No. Nasal cartilaginous capsule. And on the surface, you have the developing nasal mucosa, similar to the slides that we checked. So you see developing nasal mucosa on the surface. That's number 6. You might even see developing nasal glands. So developing nasal mucosa. So there's some glands. In the slide, you can actually see nasal gland development. Which part of the nasal cavity is growing here? No, it's hard to say. It might be the olfactory part, it might be the respiratory part, but you don't need to distinguish the part. Just nasal mucosa development, it will be okay. Next, let's move here to the back of the skull. So if we move a bit back, so this slide is very big, you will start to see brain development. Brain development. Now, sorry, it's not 6, it's 7. So 7 will be brain development. 7's brain development. Go to number 7. So brain development. So you don't need to distinguish parts of the rat's brain. We have brain development slide. In that we check different parts of the brain. You just need to remember, so brain development, that's all of this here for the rat. Brain development. That's number 7. And what else we can spot? You will spot that there's some bone tissue development on the top, here on the surface, number 8, desmocranium. So desmocranium. I will show that also on the microscope. So next to the brain, you might see desmocraniums. Here, this flat area with small bone fragments, that's desmocranium. So these small bone fragments are developing desmocraniums there. So desmocranium, small bone fragments with the green arrow, as you can see, that's desmocranium. And if we have desmocranium, if we go to the opposite side, we will see number 9, chondrocranium. Chondrocranium. It's on the opposite side here. You will see a plate of hyaline cartilage, maybe some fragments, but chondrocranium can appear. So chondrocranium. So if you see hyaline cartilage, that's chondrocranium on the opposite side. Here there's a big plate. On this slide there are small fragments. In some slides you might see even inner ear development, like in the microscope. We can actually see the developing inner ear in some areas. Here it's not that nicely visible, some fragments, but chondrocranium stands out a bit. So you see chondrocranium, so plate of hyaline cartilage, chondrocranium. Okay, next. You can spot here, number 10, developing vertebral column. So number 10, developing vertebral column. And I will move in the microscope also. Let's move along. If you see these developing vertebrae, then vertebral column development, there are ossification centers, some spinal ganglions even, as you can see, but developing vertebral column is visible. And in some slides, inside the vertebral column, you might see spinal cord development. You can see it in the slide picture, developing spinal cord, number 11. In the microscope, as I'm checking here, we don't really see the spinal cord at all. In the microscope, we don't see the spinal cord that well, but we see the spinal cord in the slide picture. Spinal cord can appear. Let's go towards the back. So behind the spinal cord, you will see, so number 12, skeletal muscle tissue development. So you will see skeletal muscle tissue. Typically close to the spinal cord, you will see long fibers, sometimes in cross section, but skeletal muscle tissue can appear. So developing skeletal muscle tissue. But what you need to be careful with, I will specifically encircle these fragments here. You will see very big fragments of brown adipose tissue, number 13. Between muscle tissue, you can have brown adipose tissue fragments. Brown adipose tissue. In the slide, these brown adipose tissue fragments look like this. You see They almost look like the liver tissue a little bit. And if you zoom in, again, you will see lipid droplets. If you zoom in, it has a very similar look like in dog's head slide. So you see small cells, almost looks like liver tissue, but this is brown adipose tissue. You have small lipid droplets inside, brown adipose tissue. So brown adipose tissue can be found on the back of the rat's embryo, large quantities, at the back region, next to muscle tissue. And so here below, this is muscle tissue and brown adipose tissue on top of it. So that's the tricky part. Sometimes it can be easily mixed up. Okay, what else? Let's go back to the neck region. Let's move to this area, number 14. If you check the lower jaw, I will move there. So I will move back to the tongue. Tongue was here. You see there's a big tongue. And if we go below the jaw, you will see a big gland. Just write submandibular gland. So this is developing submandibular gland. So developing submandibular gland. So it's a very big gland. Technically, sublingual gland also can be here, but in most slides, it's just the submandibular gland. If you remember this in the rat slide, it's more than enough for this area. Then, what else is in the neck area? Here, number 15, you see small plates of hyaline cartilage. I will check here also. Here in the neck region, you see there's some plates of cartilage just behind the tongue. The tongue is here, and you see some hyaline cartilage plates. Developing larynx. So number 15, developing larynx. And beneath the larynx, you see there's some glandular tissue also next to the larynx. If you zoom in here, you see rounded structures with a pinkish fluid inside. Developing thyroid gland. Number 16, this is developing thyroid gland. It's not in all slides, but sometimes can appear next to the larynx. Developing thyroid gland. So you see the thyroid gland. In high magnification, you can see developing follicles. But developing thyroid gland. So far, so good. Be careful. You might see brown adipose tissue also in the neck. It's not in the slide picture, but you can easily spot it here. Next to the thyroid gland, even some brown adipose tissue can appear. Okay, we have checked the head and the neck region. Now let's check the thoracic cavity. You have to spot this structure, number 17, developing thymus. This is a very helpful structure because if you see it, you know that you are in the thoracic cavity. It's very basophilic, so developing thymus. Number 17, developing thymus, so highly basophilic structure, and tells you that you're in the neck region. Beneath the thymus, here you see heart development. So there's the developing heart development. You don't need to distinguish the parts of the heart, but you see heart development very well. So heart develops below the thymus. You see the heart. It's quite big in this slide. You don't need to distinguish the blood vessels and parts of the heart, just heart development. And next to the heart, as we already know, we have lung development in pseudoglandular stage. So if you see lung tissue, number 19, lung development in pseudoglandular stage. Lung development in pseudoglandular stage. So we see lungs very well. If we move in front of the thymus, and if you see hyaline cartilage in front of the thoracic wall, like here you see these round fragments, rib development. So this shows rib development. Rib development. So 20 developing ribs. So developing ribs will rib development. So ribs develop here. Okay. What else? Back to the thymus, and now let's go closer to abdominal cavity. Between the abdominal and thoracic cavity, you will see skeletal muscle tissue. So this thin plate here with number 21, developing diaphragm. So 21, developing diaphragm. So diaphragm development. So developing diaphragm, you will see it as a plate of muscle tissue. Developing diaphragm. And behind the diaphragm, you see a very big organ, liver development. So number 22, liver development. If we zoom in this liver, we can find the liver cells. So liver development, liver development. Liver development. Liver is massive. If you see the liver, you're in the abdominal cavity. So all of this is the liver. So very large organ, fills the whole abdominal cavity. Below the liver, you see developing small intestines. So small intestine development, that's 23. Again, if we zoom in, we can add small intestine structures from the slide. And what's also good here, that's 23. I will clear these drawings. So 23, small intestine development. But as an extra, you will see some eosinophilic tissue. That's 24. In the slide that's here, developing pancreas. So developing pancreas. So you can see some pancreas tissue in the slide. So developing pancreas. We would zoom in. So we can spot the pancreas. So this pink eosinophilic tissue, this is pancreas development. So developing pancreas. Then what else? If we move below the intestines, you might see a larger cavity. Here there's an organ with flattened walls. You can see it here also a little bit, 25, developing urinary bladder. So urinary bladder typically was much thinner than the intestine. There's smooth muscle tissue, thin epithelium. So developing urinary bladder. And then what else? Very rarely, but sometimes you might spot developing, not developing, but you might spot umbilical cord tissue, umbilical cord even. Umbilical cord. In this slide, it's not a guarantee that we will see the umbilical cord. No, we see small fragments of it, so umbilical cord can also appear. So, that's the idea, yes? We will see it. I will show it here. So, umbilical cord. What else? Some structures that can appear in addition. So let's check the back wall next to the vertebral column. So I go behind the liver. And here we see our old friends, metanephros and the adrenal gland. In the slide, it's also here, 27. Metanephros and 28 adrenal gland. It's not in all slides. So there's different slide variations. So the idea is you need to understand the anatomy in general. So if you know topographical anatomy, where each organ should be positioned, you won't have big problems with this slide. Of course, it's a rat, it's not a human, but many organs are located in similar locations. And if you understand that, then it will be more than enough to understand the whole slide. My recommendation is to find the following when you check the slides on your own. Where the oral cavity starts, this area, thymus, and intestines. When you check the slides, try to find these points. The oral cavity, so the start of the mouth and the lips, thymus, and intestines. If you see these three areas, you can understand the regions. So if you see the thymus, you know that you're in a thoracic cavity. If you see the intestines, you're in abdominal cavity. And if you see the start of the mouth, the tongue, you're in oral cavity in the head region. If you understand this, then you can navigate through the slide. So these are the three areas you need to emphasize when you learn and to try to check the slide on the microscope. So this is very important because these are gigantic slides. So these are very big and very tricky slides to check. So we have 3 minutes left. I will briefly show you some variations of rat embryos that look a bit different than the others. If you want to take clean shots, this is your chance. If not, I will very quickly show you some variations. I won't go through all the structures that are possible here because different rat embryo slides can have different structure variations, but we can try. So let's check another rat here. What stands out in this rat? You have the inner ear development. So you see the inner ear development. That's special for this rat. And what is also special, no intestines, very big liver. No intestines in this rat, so very big liver. So this is a big liver. And behind the liver, you see the pancreas, metanephros, and adrenal gland. So P, pancreas, and metanephros, A, adrenal gland. So this is a very large slide also, but the tricky part is about the structure. So there's a bit different variations. So there's the inner ear, you can spot, very big liver, pancreas, metanephros, adrenal gland. No intestines here. So that's special here. Then another variation. Let's take this one. Let's check the neck. So if I zoom in the neck region, in this slide, that would be here. I will check this area here. You will spot something interesting. So here, this is the larynx. I will put L or LA larynx. This is the thyroid. And this is the esophagus. In this slide, you can actually see the esophagus. You see very thick epithelium, esophagus. So esophagus. Esophagus can appear in the slide also. That's unique here. And also what's unique, you see the aorta. You see this big artery that goes in front of vertebral column, the aorta. Here you have the dorsal aorta, developing aorta. So that's special here. You can see the aorta in the slide. Then what else? Not this one. This also, okay. Here the head is a bit damaged, but the structures are similar. Here, what's interesting, you see the foot of an embryo. So this is the leg of a rat's embryo, and the lower jaw is pressed towards the stomach. So you see the chin or basically lower jaw is pressed towards the anterior wall. So this embryo is folded a bit, but still you have the oral cavity, the thymus, and the intestines are here. You can use it to navigate through the slide also. That's special here. And what else? Again, this slide is special because you have the eye. So there's eye development even in this slide. Eye development. So eye is here, and also the ear appears here. So the inner ear development. But again, some of these variations you will need to check on your own. We are out of time, but thank you very much. So I have a consultation this Thursday, so if we want to check some other slide variations, we can discuss them during Thursday's consultation. If needed, we can arrange a consultation at the start of the next week, even, or if needed, if the times or regular consultation not available. So thank you very much. See you later. Good luck in the colloquium. Yeah. My consultation 16:30 in the afternoon, so 4:30 in the afternoon on Zoom. Zoom links are in the studies. Okay, colleagues, thank you very much.